All right, grade 12s, we're continuing on in our discussion regarding mitosis, which was our last lesson. So in this lesson, we're going to talk about cancer, and we're going to talk about cloning, and we're also going to talk about stem cells. So that's what we're going to cover here, and I'm going to go through this really pretty quickly. So cancer cells are cells of the body that are simply dividing at an uncontrolled accelerated rate. They're undifferentiated, which means they're not specialized, which means they're not going to do the job that they're supposed to do. And then, of course, that creates a tumor, and that tumor impairs normal function. Metastasis is when those cancerous cells can break free from the area that they find themselves in, usually either in the bloodstream or the lymph, and then they can relocate to another part of the body. This is just a slide, guys. As we review mitosis, you should be able to pick out the stage or phase of mitosis that you happen to see in a diagram. Sometimes it's a little tricky when they're real cells from real life, but and especially metaphase might be difficult because sometimes the visible chromosomes there have very long strands to figure out. But I mean, we could probably find that, hey, here we're probably looking at a prophase example, and maybe metaphase here, if we consider this to be the equatorial plate. They seem to be congregating around the middle. Here we've probably got telophase, right? And we've got two nuclei, if this was one cell, and we can see that the chromosomes are probably unraveling to become chromatin. We're going to get our nuclear membrane reforming there, and then cytokinesis is going to occur. These are plant cells, so we're going to get a cell wall to create two brand new smaller cells. Now, let's turn our attention to cloning. All sorts of things surround cloning, a lot of science fiction kinds of things, but let's get our definition for cloning. So our definition for cloning is the process of forming identical offspring from a single cell of tissue from a parent, and then the clone originates from that single parent cell, and both the clone and the parent would have identical nuclear DNA. It says, therefore cloning is considered a form of asexual reproduction, and vice versa. Asexual reproduction is a form of cloning. Now, very quickly, you've heard these terms from even middle school science somewhat. Let's go through this very quickly. Binary fission is when we might have a bacterium that's able to divide into two. That's called binary fission. Or budding, where a parent has a growth called a bud that is essentially a genetically identical to the parent, and it will grow until it eventually is able to liberate itself or separate from the parent. In the case of an organism called a hydra, that's a good example. Or even strawberry plants, in a sense, grow runners, and that's a form of budding. Or spore formation, in the case of molds, for example, is another form of asexual reproduction, where those spores can tend to be a genetic replica of the parent. Now, let's turn our attention to twins. There is actually a situation where identical male twins married identical female twins. Sure, everything happens in this world. Now, so in the case of monozygotic, we're going to talk about that in a second, guys. That's another term for identical twins. They're going to share, of course, and grow in the uterus together. That's whether they're identical or fraternal twins. There's the Ashley sisters. Okay, we're going to talk more about this in a second. All right, so let's talk about monozygotic or identical twins. Monozygotic means, like, literally one zygote. So at this stage, no differentiation has taken place. The cells are not only identical, but each has the potential of creating an embryo. So remember, when you're at the 2, 4, 8 cell mass stage, when you're in that blastocyst stage, all those cells are identical to one another. And if they should happen to separate, or you create two blastocysts, then you're going to end up with identical twins from that one zygote. So we can kind of see that happening here. That embryonic cell mass has separated, and each one of those will differentiate into identical twins. We're going to see this totipotent term come up when we talk about stem cells, because essentially right now, guys, these are stem cells, and they're totipotent, which means they can actually reproduce themselves. Okay? So for identical twins, there's only one fertilization event. One sperm, one egg, one zygote, and we're going to end up with 2 embryos, often only 1 placenta. Now, let's talk about fraternal or non-identical twins, or another term is dizygotic twins. So in this case, we've had 2 separate ovulation events. So it just so happens that in 1 month, maybe the one ovary ovulated 2 eggs, or maybe each ovary released 1 egg, and then of course 2 separate sperm find those 2 separate eggs, and then we get 2 zygotes, so dizygotic. We're going to end up with 2 embryos with 2 separate umbilical cords with 2 separate placentas. Now, before we talk about cloning, what did I want to talk about here? I can't remember. I was going to make one more comment in regards to the twin situation. I think what I wanted to say was that each one of those fraternal twins are not going to be genetically identical. They're going to be genetically different as any brother or sister would be. They just happen to be born at the same time, but no more genetically alike than, say, a brother and sister or brother and brother born at different times to the same mother. Now let's turn our attention to animal cloning, guys. Now we're going to do this very quickly. I might actually skip a couple of slides. I'm going to get to, let's take a look at this slide to kind of talk about cloning. So animal cloning was made famous, I think, back in 1996 when a mammal was cloned. And so essentially what happened was we have this mother sheep and some donor cell, and it's from her udder. They take some donor cells, a typical body cell, and they're going to take that cell with its nucleus, and they're going to fuse it with an egg cell that is taken from another mother sheep, female sheep, and they just take that egg cell, which is unfertilized, they remove that nucleus, they then fuse the nucleus of this body cell from that sheep. This is the sheep that we're attempting to clone. So we have all its genetic information in this nucleus. We fuse it to make essentially a zygote here with one cell with all the genetic information from this sheep right there. Then they use some electrical razzmatazz, and they're able to cause it to divide. So to become, as we have in the blastocyst, an embryonic cell mass. They let that grow to a point where they can then put it into a surrogate mother sheep to simply carry that baby to term, kind of like an in vitro fertilization situation. So this could be any mother sheep. And then when the baby is born, this is a cloned lamb that is genetically identical to that sheep right there. Okay? So that essentially is how cloning a mammal works. Okay, there's a few more examples in the PowerPoint that I'm not going to work. Don't want to spend too much time on this, guys, because again, I have to pick and choose with our limited time for me to go over some concepts. I'm picking and choosing a little bit where to really spend our time. Now let's just end this video by talking about stem cells. So stem cells, which it's not directly cloning, but stem cells have the ability to become different kinds of cells. Remember we talked about this term totipotent? So totipotent cells are able to copy themselves, but they can essentially become a whole nother individual, like in the case of identical twins. But we also have cells that are called pluripotent. Now pluripotent cells are cells that have the ability to become many different kinds of cells in the body. For example, the stem cells that are in bone marrow, they in a sense are in a form, in a way pluripotent. They can become any kind of blood cell, red blood cell, all the different white blood cells, and platelets. All right. So again, the idea is that if we can kind of harness the ability to take an individual's stem cells, we could create tissue that they would not reject because it would be their own stem cells, which would be a real step forward into that. All right, now embryonic cells from the umbilical cord are, you know, in a sense, depending on your view on abortion, that's where a supply of stem cells could be gotten from. And of course, that creates a lot of ethical issues that there's lots of discussion surrounding. All right, so that ends this video. The next video is going to get into meiosis.